This is a production of Cornell University.
So last year I left off telling you guys about our efforts to map large effect early blight resistance loci in 2 tomato breeding programs. And I was telling you how we plan to use this information to learn more about the tomato early blight pathosystem and to develop new inbreds with enhanced disease resistance. So I showed you that we found 4 early blight loci of real interest here, 2 of which were predictive of stem lesion resistance, and that's EB 1.1 and EB 9 here, colored in green. And 2 were predictive of resistance to defoliation, and those are EB 1.2 and EB 5, colored in purple. Now, of these 4 QTL, EB 5 and EB 9 had statistically significant resistance effects across 5 breeding populations over the course of 3 years, while EB 1.2 was more inconsistently detected. Now, we've since used this information to inform a genome-wide marker-assisted backcross scheme to rapidly pyramid these early blight QTL into 2 elite tomato backgrounds from 2 separate market classes. Now, the first of these releases, CU 191357, is a Northeast adapted, medium-sized fresh market slicing tomato, and the other, CU 201041, is a determinate Midwestern processing type. And these tomatoes, which carry additional resistances to a host of other diseases, are being thoroughly evaluated this summer and are being released to third-party breeding programs through the Cornell Vegetable Breeding Institute. Now, alongside these inbred releases, we also wanted to provide reliable and background-independent markers for these early blight resistance loci. And the traditional way to do this would be to iteratively find map, refining the confidence intervals until it was possible to identify and validate confidence or candidate genes that would become the target of molecular markers. But this kind of work is really time and resource intensive, especially when it comes to quantitative disease resistance. And from a practical breeding perspective, we don't really need to know the causal variants, and I would argue it's good enough just to find the smallest extant resistance-bearing haplotype or the smallest introgression from the ancestral resistance source. And then from there, you can design markers that are specific to that haplotype, and these markers should be capable of predicting resistance across breeding programs, regardless of the recent ancestry. So my project partner and I are attempting to do exactly that by sequencing landmark resistant inbreds from more than 80 years of early blight resistance breeding, and by applying the tools of population genetics. So to do this successfully, our first challenge was to dig through the available breeding records and to come up with a hypothesized ancestral pathway for each of our resistance QTL. And I want to take you now through one such history, which begins with a tomato breeder by the name of Dr. George B. Renard. Now, Dr. Renard was an avid bird watcher, and in fact, he served on the board of the Cornell Ornithology Lab, where he helped them to build out their recordings of the US and Caribbean bird songs. But in the 1930s and 1940s, Dr. Renard worked at the Vegetable Breeding Laboratory in Charleston, South Carolina. Now there, he and C.F. Andrews worked out the first early blight inoculation protocols, allowing them to screen tomato accessions for genetic resistance. And he and Andrews found resistance in several tomatoes, but one, an English forcing tomato by the name of Devon Surprise, is described in their 1944 publication as having particularly strong resistance to early blight stem lesions, which is a phase of the disease also known as collar rot. Now, importantly, they also observed inheritance ratios that suggested that this resistance was caused by a single underlying locus. Now, Dr. Renard eventually left the USDA to become a tomato breeder for Campbell's. And the story might have ended there, were it not for another important and more well-known early blight-resistant tomato called Campbell's 1943. Now, this tomato was described in a 1977 publication by Thomas Barksdale as having excellent early blight collar rot resistance. And Barksdale also writes that, quote, Campbell's 1943 was kindly provided us by Dr. George B. Renard of the Campbell Institute for Agricultural Research. So this suggests that Dr. Renard, having previously discovered the early blight resistance in Devon Surprise, then used this tomato to create Campbell's 1943. And from this point forward, recreating the pedigree is much easier because Dr. Randy Gardner, now emeritus professor at North Carolina State University, picked up Campbell's 1943 and used this as a source of resistance for his 1980s release of NCEBR2, which had early blight stem lesion resistance. And then NCEBR2 was used in a subsequent 2008 release of NCCELBR1. And then NCCELBR1 was used by the Cornell tomato breeding program as a source of resistance for CU151095, which is in turn in the pedigree of our two newest tomato releases. And if we repeat this process for other sources of early blight resistance in the pedigree, I was able to put together this greatly simplified hypothesized pedigree of early blight resistance in our tomatoes that spans some 80+ years of breeding. Now, the next step is to obtain whole genome sequences for all of these lines. Now, some of these sequences were available from the NCBI Sequence Read Archive or from our collaborators at The Ohio State University. And the remaining 9 tomatoes, though, we sequenced ourselves. We built libraries using the NexteraFlex protocol and then ran them on a NextSeq 500 sequencer. Reads were then aligned to the Heinz 1706 reference genome, and this is the 4.0 reference genome. And SNPs were called with the GATK4 pipeline, and this yielded about 12x filtered coverage over the variants. So again, if our hypothesized pedigree is correct for the chromosome 9 QTL EB9 coming from Devon surprise, then we should be able to observe a shared haplotype near EB9 in Devon surprise and in all of our landmark early blight resistant inbreds. Now this plot, which is based on the output of the ABBA test, shows us that this is indeed the case. Now, this analysis looks for the evidence of introgressions in triplet SNP contrast, giving a D statistic that can be used to paint chromosomes by ancestry, and also to visualize homologous haplotypes. And what we find is that in all of the early blight resistant entries, they share a solid red block of SNPs near the EB9 locus. And this suggests a shared Devon surprise introgression. In contrast, SNPs that are more consistent with a Heinz 1706 ancestry are colored in blue. Now, another way to find regions of high sequence similarity is to window the genome and then to use a clustering algorithm to intelligently group the resulting haplotypes. Now, here you can see an example where I've projected 30 tomatoes from 4 different clades onto a 2-dimensional PCA space for a single genomic window near the EB9 QTL. Then I used the k-means algorithm to cluster the samples and found that it perfectly separated the early blight resistant and susceptible large fruited cultivars from each other, from 2 cherry tomato types, and from the wild species Solanum haverchides. Now, this type of unsupervised clustering analysis is really nice because it's computationally pretty trivial and because we can improve our ability to distinguish among observable haplotypes as we add additional sequences to our database. Now, once a sample in a window is classified as a member of a group, we can then ask if that sample is in the same or different group from some putative ancestral line of interest. We can then color the areas that have the same k-means cluster designation to visualize homologous haplotypes and even introgressions. Now, here is one such plot of chromosome 9, inclusive of the 8 resistant samples that I talked about earlier. They're blocked by this green resistant bar at the top, and then 2 random susceptible samples from the pedigree I described earlier at the bottom. And the genomic windows were colored if, in any window, they were in the same cluster as Devon surprise. And the resistant tomatoes are also sorted according to their hypothesized ancestry, with Devon surprise, the putative donor, followed by C1943, NCEBR2, NCCELBR1, and then our 3 most recent Cornell releases. And what's really cool about a plot like this is you can actually watch the haplotype from Devon surprise decay over time with phenotypic selection, going from a large introgression in Campbell's 1943 to a relatively small one in the North Carolina tomatoes. And here on the right, we've zoomed into a 95% confidence interval for the EB9 locus, The black ticks denote the confidence interval boundaries and the most predictive SNP marker at the center. And we can clearly see that the smallest contiguous Devon surprise haplotype was present in the sample NCCELBR1A. And this gives us a new resistance haplotype size that's about 33% smaller than before, or about 1.2 megabase pairs. And furthermore, these data strongly suggest that our modern early blight stem resistance traces back directly to an early 20th century tomato developed in Devon, England, which was then improved by Dr. Reynard and through significant public sector breeding efforts spanning more than 40 years. Now, I don't really have time today to tell you the whole story for the other resistance loci here, but I did want to show you that we can use the same methods to trace our early blight 5 resistance locus to an unimproved tomato breeding line named Hawaii 7998. And in this case, we were actually able to shrink our interval size by more than 60% over our prior boundaries. So this demonstrates the power of this approach to reduce QTL confidence intervals without ever having to do fine mapping. So now that we have some preliminary success with this project, we want to scale this up to include wild species and many more modern and heirloom tomatoes for which archived sequences already exist. And this should allow us to further trace the history of our resistance loci back to the wild species donors, and also to determine whether there are other sequence tomatoes sitting in the gene bank that share this early blight resistance haplotype. We aim to identify SNPs that are specific to this resistance haplotype, to find excellent candidates for portable and background independent molecular markers. And finally, we hope that this work will help to reveal tomatoes with unique versions of the resistance haplotypes, which could predict the presence of novel alleles conferring even stronger early blight resistance. And with that, I'd like to thank in particular Martha Sutterman, who's my project partner on this. This was largely funded from a SIPS Schmidt-Novak Small Grants Program. I also want to thank from the Missouri Lab Kyle LaPlante, who helped us to brainstorm these methods for our project. I'd like to thank everyone in the Mushler breeding program and in the Francis breeding program at OSU, and also my graduate committee members. And with that, I think I have a couple more minutes here to take questions. Stop sharing the screen.
You can type if you have questions. Can take 1 minute for just 1 minute for question. Running out of time.
Any leads on the background of Devon surprise? So the trail stops there. I tried looking up, so I was able to find the company that sold Devon surprise, but they've folded long ago. I can't identify who the breeder is. So I think from this point forward, we're going to try and find a wild species that best supports introgression and Devon surprise, and make our best claim that that's the ultimate source of this resistance. Let's see. Like the historical approach, thank you very much. Do you think that on reducing the haplotype sizes for finer mapping, you could be confounding the effects of other potential causal QTLs? Do I think that reducing the haplotype sizes for finer mapping? I'm not sure I totally understand the question there, but I don't think that we are confounding the effects. I think we could be leaving out other QTL. And in fact, I have already left out several QTL that were of minor effect here. We really wanted to focus on the largest QTL of interest, largest effect QTL of interest, because that's really what the breeding programs are most interested in when they're trying to bring these into their background. And then another question, what tool were you using for haplotype visualization? So I used Python for creating to do the k-means clustering analysis. And then these plots are actually based on, they're actually just ggplot bar plots in which I've stripped out all of the unnecessary background detail, all of the grids and everything, and used facet wrap to stack all of the entries on top of each other so that we can do comparisons across entries.
Nice.
Okay. I think there is one more question.
Do we have markers for QTL5 and QTL9 that seed companies could use? Yes, I mean, we currently have markers that we are using in our own breeding program, but really the purpose for, the motivating factor for all of this work is to define markers that will be useful for all backgrounds. So yes, we currently have markers that could be used. They may not be polymorphic in all backgrounds, but we hope that by the end of this work, they will be robust enough for any and all programs to use them.
Okay. I think there are no more questions. Let's thank Taylor for your nice work and move on to the third speaker.
Thank you.
Yeah. Can you
This has been a production of Cornell University, on the web at cornell.edu.